Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now a part of the Finding Genius Foundation. Today I have Manuela Ramalho. She's a cell and molecular biologist in the Department of Ecology and Evolutionary Biology at Cornell University. And we're going to talk about ants and some interesting features about them. So, Manuela, thanks for coming.
Thank you for inviting me, Richard. It's a huge pleasure for me to be able to talk about these topics that I love, ants and microbiome.
Oh, good. Yeah, I've been looking for people that deal with the microbiome of ants. And it just so happens that my kids always leave food in their rooms, and so we have ants now that the weather's warming up, like swarming all over the place. And I don't know what kind they are, but I'm like, ugh.
Yeah, so I understand you. I have kids as well and dogs. So yeah, ants love them. And I grew up in Brazil as I'm Brazilian. So in the tropics, we have ants everywhere.
Well, how did you get interested in them? Just because you grew up around them and you were curious, or what's the story?
So yes, as a Brazilian, I grew up in the tropic with a lot of insects around, but was just in the college that I met my advisor, Dr. Odair Correa Bueno, and he was fascinated by ants. So I was starting to do my researchers as an undergrad, and more I learned about them, more I fell in love all of these ants facts. But specifically, speaking specifically about microbiome and symbiotic interaction, that it's a big part of my research today, I was first fascinated with Wolbachia and ants interaction. So Wolbachia, it's a very common bacteria in many insects, including ants. And we still know very little about the effects of this bacteria and what they can cause for the host. So this whole unknown world of symbiotic interactions with microorganisms and insects caught my attention. And with each study that have been published, we are discovered that there is no one single standard or a single rule that can apply for the entire system. So these symbiosis can be variable and complex, which make our tasks challenging, but also super fun. And definitely, that's why I'm very interested in continuing studying this.
So you're focused on the microbiome of ants, or what's your particular focus?
Yes, I'm actually, to understand the patterns of biodiversity, we must consider symbiotic interaction as they can shape the animal evolution. So I'm interested in questions that how we can change this symbiosis, how can we explain all of these diversities? So these kind of questions that intrigues me and I love to study. But my researcher focus on understanding the mechanisms that impacts the microbial communities, unraveling the role of the ecology, behavior, stage of the development, diet, and also the phylogeny of the host in these symbiotic interactions. And then to answer these questions, I use ants as a study model. And it's very known for several ant genera that symbiotic interactions with microbial community have a profound impact for the host. So more than that, ants have a wide distribution across the globe, and their immense diversity and behaviors and ecology And also, I think they are beautiful and they are fascinating. So this was a perfect combination for me.
Okay. So what's have you sequenced the microbiome of various ant species? Like what have you done in terms of the microbiome of ants?
So we already studied several different species, microbiome of different species, actually in different parts of the world. So different behaviors and everything. And one of the most recent studies published last year, one ant that it's called Dasyton ermisium. It's a predator who lives in the neotropics. And when we sequence their microbiomes associated. So for that particular project, we want to also test that the role of the diet for that specific species because they are predators. So what is fascinating about this study is my collaborators was able to collect the entire colony that was sitting there, living there in a rainforest in French Guiana. So they bring this colony to the lab, and we feed them with a different diet for a long time because we want to expect to see some difference in their microbe. But was surprising to all of us that even after a while in a different diet, keeping a lab, we found no change in their microbes associated with these ants. And this was very huge thing for me because I thought that if you're changing the diet, we are changing everything, but at least not for the workers of this species, Dasyton, that we are working on. So this is also was interesting because made me think that now I want to see if this pattern will continue the same, if we test and made the same experimental and test the effects of the diet and the larva of this species. So as I say, there's no one rule, and we are still learning a lot about like how can we change and understand the microbiomes associated with ants, but actually to everything, including us.
Well, what have you found with ant microbiomes? I mean, what's interesting or unique about them?
So, for example, one thing that I like to study microbiomes associated with ants, it is ants is a group like who live in society. They live inside of a colony. They can live alone. So it's a good system for applying questions like how the social interactions can affect the microbiome. And if you were thinking in a big picture, we can apply several of these models and questions for even our species, right? Because we still, and now in pandemic, not so much, but we also live in society.
Well, okay. So, I mean, again, what specifically are you observing in ant microbiomes? You know, like, for instance, I know that ants will culture fungus sometimes. So do you see that fungus appearing in their microbiome, or there are bacteria in them or fungi that work with the fungi that cultivate? Or, you have, I guess different predators that prey on ants. I mean, you have some ants that are venomous. Do the venomous ones have special microbes that seem to produce the venom for them? Like, what have you discovered?
So everything that you say, probably there's a lot of people working in all of them. So, for example, we have in just one ant that I'm working right now, just one single ant, I found more than 1,000 different bacteria. And I'm sure that the sequencing, it is good, but definitely not getting the entire diversity of bacteria associated with them. So, for example, we know for other species that they have also fungus associated, and could be that this fungus could be like food for ants, but could be also this fungus could be pathogenic for the ants. So that's very like depend of the ants that we are talking about, depend of the system that we are applying. And that's the entire like unknown microbiome that we're still discovering. And for sure, several of the species of the bacteria and fungus that we are discovering associated with ants, no one ever discovered yet. So we still have a lot of things like to work in the future, considering like what is the function of all of this diversity of bacteria and fungus that we are discovering. So that's another level of years and years to be studying for other people to development. So we still are just like this technology that we have available now. It is great, but it's still very limited for answer all of these amazing questions that we can think about it when we are talking about insects and microbiome.
Have you tried looking at ants in different conditions to see as their microbiome changes? If you look at the microbiome of the queen versus like the workers or the drones or whatever you call them? What does that look like? I mean,
Yeah.
I'm sure there's tons of things to see. What have you observed?
Yeah. So, for example, we know one ants that Campanotus ants, they're famous to have one specific bacteria that called Blockmania. We have opportunity in my PhD to collect like entire colonies, see inside of the colony, all of different castas that was available there. So we compare the microbiome of inside of the queens, inside of the eggs, inside of the workers, and the larvae, the males, and we are able to see like how they are similar with each other between like inside of the species level, but also could be a very like similar inside of the colony. That means that the ants for this species, they are interacting so much with each other that it's able to kind of keep all the microbiomes pretty similar inside of the colony.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
So that was pretty fascinating. But in that specific paper that we work on, we have the opportunity to see that the larva has a great diversity compared to the other castas inside of the colony. And that's, we think this is an answer because the larva stage is where the ants are feeding, are eating a lot, right? Because they need to be like all of this food for the next development stage that it's pupa, where they are not eating anything for a long time until they are born. So in the larva, they have more food, and that's probably because they have more diversity of microbiomes associated. But that's pretty much what we know about, like, at least these specific species of ants. We still have like more than now we know so far that we have more than 13,000 of different species of ants in the world, and millions of different bacteria. That is probably some of them bring a bunch of benefits for these ants, and some of them, these bacteria could also be prejudicial for these ants. So we still have a lot of things to learn about the entire system.
But if you compare amongst ant species, since there's so many of them, I would think that there would be common, bacteria that are in most of them, and some that are unique to only certain species. Have you been able to observe that?
Yes, I think yes, there's some bacteria that it's probably like related to just one species, and that's what we are seeing so far. We do know that there are some bacteria that it's more broadly for not the entire, all the ants that we know so far, but for several other species, not necessarily related ants. So, for example, Rhizobialis, we know that Rhizobialis is it's bringing especially nutrition benefits for ants, and it is not associated with just one specific genus of ants, that it's more broadly thinking in phylogeny perspective. But still, we don't know, there's no just one bacteria that it's just happening in the entire, all the ants that we know so far. Not that we know yet, but maybe next year, maybe tomorrow.
Are you able to figure out the metabolites that the bacteria you found produce that work with the ants, that are part of their symbiosis, I guess, with the ants? Have been I know sequencing is great, but if it's 16S, I don't know if it tells you much, even if it's shotgun. But then it doesn't tell you about the metabolites they may share back and forth and what the role is. Have you been able to figure out the role of any of these bacteria? And again, what substances they're doing? That's
an excellent question. So for my studies, I'm focused more in the amplicon sequence, so the 16S, as you say. So my question it's for now answer more ecological questions. So we are not focused in the whole metagenomics, shotgun metagenomics, but for ants, other collaborators or the friends of mine, they were able to discover that, for example, turtle ants, that it's called like genus Cephalotes, there is some bacteria that able to increase the nutrition of the ants by recycling nitrogen. So, and there is these bacteria bringing these benefits for the host. And they did that exactly using the whole shotgun metagenomic to discover. So there is some bacteria that people are discovering that there is benefits, but there is still few studies that was able to actually demonstrate it, what is the function. And for example, for Wolbachia that I already mentioned before, we still there's just last year that there's 2 studies that show that Wolbachia could accelerate the cycle of the colony.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And other people for other different species just show up that Wolbachia could be bringing also nutritional benefits in terms of vitamins for the host, but yet, this is actually happening for the entire ant family, or this is more related for some specific strains of Wolbachia? We still don't know, and we still have a lot of things to discover.
What is Wolbachia, by the way?
So Wolbachia is one of the most common bacteria that we know that it's associated with insects. But for example, for ants, besides this bold study that I mentioned before, we still don't know nothing what its effects for the host. And that's fascinating, like how one of the most famous bacteria associated with the ants, with insects, we still know so little about the effects on the host.
Yeah, I'm interested again, like in bullet ants. I know that they, I guess sting and really cause people a lot of pain. But what does it, the bullet ants? Have you looked at them, for instance, to see if they have unique bacteria that facilitate the venom?
Yeah, I didn't have opportunity to look for those.
How much would that cost, by the way? Would that be very difficult or take a long time to do to do a targeted sequence like that on bullet ants?
I don't think it is much expensive because this technology is now it is getting less and less expensive over the years. But I think it is also depend where you work, like what is your facility. Like I know inside of Cornell, we have some discount for people here. So I'm not sure if this price will change in a different states, but it's not so expensive as it used to be when I was in Brazil in 2017. So here in US, it is not that expensive. Probably we're spending more collecting them.
Interesting. Well, what experimentation do you want to do? I mean, what signals are you getting or what interesting data are you getting so far from the sequencing you've done?
So now I'm focusing a study with Cephalotes, the turtle ants that I mentioned before. And I want to use well-supported phylogeny to see the signal for the microbiomes coevolution associated with all of this diversity inside of Cephalotes turtle. So hopefully soon we will be submitting this paper, and we are trying to see now in a different way, like every single groups of bacteria can have a different life history, and some of them could be co-evolving, but not all of them. So let's see. Stay tuned.
Again, what are you trying to figure out in terms of coevolution? How would you determine that a bacteria has co-evolved with a given ant? What would you look for?
That is a really great question. So one thing that, because we have a well-supported phylogeny of the host, so we are able to actually map how this diversity of ants could be like combined with the diversity of a bacteria inside of this. And so if we try to explain this more a simple way, basically what we can try to measure is the phylogeny of the ants will be pretty much like mirror the phylogeny of the specific bacteria that we are looking for. So that means that if bacteria, it is evolving together with the host, if they have similar phylogenies, like it's mirror one. So that means that probably these bacteria was acquired a long time ago in the evolutionary scale, and they are changing together over the year.
Again, how would you expect them to change together? Like if you go into summer or into winter, based on the dynamics of an ant colony, like again, let's say they like to harvest fungus in the summer. I'm just making this up. Have you done any studies where you sequence them at the beginning of the summer and the end of the summer and see how their microbiomes change before and after the fungus harvesting, and what does that tell you? Like, what would you do?
Yeah, so depend of the project that we are doing, the question that we want to answer. We have several flavors how different species or different questions could change what we expect, right? So for cephalotes projects, we are not expect to see changes in like summer and winter. But if you want to see the effects of changing temperature or seasonality, we can definitely do experimental with that. And I know that there are some people that are using these kind of questions to apply for climate change. So they are seeing like if we change the temperature, we are changing the entire bacteria, the entire microbiome in that specific organism. And that could be means that like making predictions for the future if this climate change happen like and change what we have, how the ants or the insects are used to right now, we can probably this could mean that we are like declining even more the insects that we have, and we have so many benefits that these insects could bring to us, right? So I think it definitely depends of the questions that you're going to do. There's a plenty of microbiome questions that could be applied.
Do you see any assistance or help from people that are looking at the microbiome, let's say of scorpions or of bees? Are there any other creatures and microbiomes that you think would be very useful to study based on what you're seeing so far?
Yeah, so I have some friends that it's working with microbiomes in bees, and they, for example, are seeing the effect of pesticides trying to change, how the pesticides could be affecting the microbiome of these associated bees. And now could be this, meaning that could be one of the reasons why the bees are being killed. So yeah, that is a really nice system and could be definitely applied for other insects as well.
What's the best way for people to find out more about your research? Where can they go?
Oh, that's a nice question. So I have my website, and I also I'm working with one of the amazing women in science and also biologist, Dr. Carmen Rowe, and she kept a very nice website and Twitter about everything that we are doing. So you can follow Carrie Moreau in the Twitter or myself as well.
Thank you. I want to thank you for coming, by the way.
No, thank you for inviting me. It was a pleasure to be here.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.